Hello, everybody. It's Miss McLean again. We have 3 pages of notes, 1, 2, and 3, that you have a student copy for, so you can fill in the blanks or write the answers on a piece of paper. And then we got 2 diagrams I'm going to be referring to. So this week, your job is to watch these notes here on the YouTube channel and then fill in the student notes. Then there's diagrams to fill in. And we will go over those diagrams here in a minute. And then you have an Amoeba Sisters video to watch. That's for Monday. And then Wednesday, you need to fill in the blank and the matching page that I've given you, as well as try the city cell analogy. We will go over those in Zoom on Thursday and Friday, as well as I'll introduce your project. And the only homework assignment you have to submit is assignment number 24, which is the cell Quizlet. All right, so we're going to go through these notes. It says the study of life involves learning about cells, tissues, organs, organisms, and ecosystems. So that's pretty much the whole class in a one sentence. Okay, so just to review again, enzymes are chemicals that speed up chemical reactions. Lipids are fats and oils found in the cell membrane. Proteins are amino acids made of carbon, hydrogen, oxygen, nitrogen, and sulfur. And then carbohydrates are your sugars and your starches. All right, so first thing is a cell. And I've highlighted in pink the vocabulary word, and in yellow are the fill in the blanks. So you can pause this at any time you need to fill in the blank. So it says cells is the basic unit of life and are so small that at least 50,000 cells can fit on this letter O. Right here on this page, 50,000 cells fit in there. They're very, very tiny. And the reason they're so tiny is to have the greatest surface area to volume ratio. If we were in the classroom, we would have done a lab with a lot of math, but we are not. So I'm just going to tell you the answer. It's the greatest surface area to volume ratio for a cell is the smallest size. Cells' jobs are to work to release energy from food, and once the energy is released, it is used to make the needed cell parts. All right, cell theory. Okay, cell theory has 3 parts. It is a vocabulary word, so it's in pink. Prior to the cell theory, people believed that diseases were caused by curses and supernatural spirits. Here we go about Halloween week, right? So if you got sick or ill, it's because someone in your family did something bad or wrong. They believed a lot in the supernatural. And it wasn't until the invention of the microscope that cells were discovered, the basic unit of life. The cell theory was developed simultaneously in 2 different places, in England and in Germany, by Robert Hooke and Anton van Leeuwenhoek, after observing the cell through a basic microscope. So basic, like really strong magnifying glass. So Robert Hooke is the person in England that actually coined the term cell. The reason why is because he was looking at cork through a magnifying glass, and it looked like little boxes, kind of like prison cells. So he called them cells. The theory was finalized by 2 Germans, Schleiden and Schwann, in 1830s, and it states the 3 parts. All organisms are composed of at least 1 or more cells, so they're either unicellular or multicellular. Cells are the basic unit of organization of organisms, so they can all be taken down to at least 1 cell. And all cells come from pre-existing cells, so we can't just spontaneously create a cell. There's no recipe for a cell. It has to come from a pre-existing cell. Okay, so today we're going to talk about the 2 different types of cells, prokaryotes and eukaryotes. Okay, vocab word, prokaryote. Pro means not professional. It means primitive. Pro means before. So prokaryotes are organisms that are cells that lack internal structures surrounded by membranes. So they have internal structures, but they don't have membranes. They're mostly single-celled organisms, things like bacteria, and they do not have a true nucleus or other membrane-bound organelles. We'll define that term in a minute. The DNA, that nucleic acid, is found in a central region in the center called the nucleoid. Vocab word, nucleoid. It's the region where DNA is kind of clumped together in the center of a prokaryote. And most metabolic functions, metabolism we talked about, is how to break down food to get the energy you need, take place in what's called the cytoplasm. So a prokaryote has a membrane on the outside. It has cytoplasm. It has a nucleoid, and it even has what's called a cell wall. And we'll talk about that here in a minute. Okay, eukaryotes are what we're talking about in these notes. Eukaryotes, U means true, so U and true. Organisms that contain internal membrane-bound structures called organelles. Organelles is a vocab word. Each organelle has a membrane surrounding it and isolates it from the rest of the cell. Okay, so again, pause if you need to write that down. All right, so today we're going to talk about all of the different structures of a eukaryotic cell. Here's a diagram of a eukaryotic cell. Lots of pieces, parts inside. It has a membrane on the outside. Okay, so a plasma membrane. All cells have them, prokaryotes and eukaryotes, including plants and animals. So you need to, by the end of this notes, be able to tell me the difference between a prokaryote and eukaryote and the difference between a plant and animal cell. Okay, so I've marked them on here. So the plasma membrane, its job is homeostasis, to maintain that stable internal environment. It is the boundary between the cell and the external environment. It provides the cell its shape. It also controls what moves into and out of the cell or enters and exits. For example, useful oxygen enters and waste products leave. Okay, so this is its job. Plasma means it's in between a solid and a liquid, and it's a membrane. It's on the outside. However, it's not completely on the outside of a plant cell, but it is on the outside of an animal cell. And its job is to maintain chemical balance within the cell, or homeostasis. So if we look at the diagram over here, this part outside of the animal cell, and then it's just on the inside right here, this yellow part of a plant cell. Okay. But plant cells are unique. Plant cells and and the prokaryotes have what's called a cell wall. It's a relatively flexible structure that surrounds the plasma membrane. It's much thicker than the plasma membrane and is made of different substances and different organisms. Things like plants have them, fungi, so fungus like mushrooms, bacteria, which are your prokaryotes, and some protists, which are single-celled organisms, have cell walls. And it's just there for further support. So if we look over here on this animal diagram, it doesn't have a cell wall, okay? But over here on the plant diagram, it has this green thing on the outside, and that's really thick, and that is a cell wall, okay? So cell wall on the outside. All right, so that is page 1 of my notes. All right, so let's come over to page 2, and it continues in yours. All right, so this one is the nucleus. It is in both plants and animals, but it's not in a prokaryote. Remember, a prokaryote has a nucleoid. This contains the nucleic acids. One of the learning targets you need to know for this unit is where are the organic compounds kept or made in the cell. So this one, nucleic acids, are found in the nucleus. And the nucleus, it controls everything. It's like the brain of the cell. It's the organelle that maintains the cell's function. It's surrounded by a nuclear envelope, which is a double membrane, 2 layers thick. Therefore, it is 4 layers total because it's a double membrane. It has 2 of those. So it's called a lipid bilayer, and it has 2 lipid bilayers. So it has 4 layers thick. So the second one again says surrounded by a nuclear envelope, which is a double membrane, 2 layers each. Therefore, it's 4 layers thick and contain pores so that materials can pass back and forth from the nucleus to other cell parts. For example, RNA is one of those things that goes through what's called the nuclear pore and can pass back and forth. Okay, it contains DNA, the master instructions for making proteins, and a long strand of DNA is a vocab word called chromatin. Chromatin is kind of like uncoiled. So for example, we're coming up in like a month or so where people are going to start decorating for Christmas. And I compare chromatin to like a big, long, uncoiled strand of Christmas lights. But when it's about to divide, the cell kind of coils that up again. And it's packed into identified what's called chromosomes when the cell is dividing. So like when you wind up the Christmas lights to store them, that's kind of what chromosomes are. So if we look at our diagram here, eukaryote, we have this big center in the middle called the nucleus, and it has pores. So here's an animal cell, and you can see it. It has these little dots, okay? So the nuclear pore is the little dots, little holes that come in and out. We don't need to talk about nuclear plasm, but the nuclear envelope or the nuclear membrane is what's on the outside, okay? And all together it makes the nucleus, and it's, again, also in a plant cell, okay? So nucleolus, we're going to talk about in a minute, is the extreme center of the nucleus. You got the nuclear pore, the nuclear envelope, and the DNA inside. Okay, so it's the control center. It's kind of like the brain, or like if we were to compare it to Fort Hays, it'd be like the principal controlling everything inside. All right, now the next one are what are called ribosomes. And ribosomes are where proteins are made. So nucleic acids are in the nucleus, but proteins are made in the ribosomes and transported later. They are found in both plants and animals, and even prokaryotes have them as well. Prokaryotes also have them. So it would not be a difference between a prokaryote and eukaryote. They're really, really tiny, okay? And the nucleus contains a region called the nucleolus, which I just showed you over here. It's the extreme center of the nucleus. And the nucleolus, vocab word, produces these tiny parts that are involved in protein synthesis called the ribosomes. This is the site where the cell assembles enzymes. Enzymes, remember, are those proteins, those chemicals that speed up chemical reactions and other proteins according to the DNA's instructions. Even though they're considered an organelle, they are not bound by a membrane. Okay, so they don't have a membrane around them because they're so tiny, itty bitty tiny ones. So I can't even really show you on here, on this diagram, where the ribosomes are. But sometimes they're located, sometimes they're free floating. I'm trying to find it. Here we go, a little tiny ribosome, itty bitty tiny, okay? But sometimes, because they make proteins, they are associated with another organelle, which is the next one we're going to talk about. And that is what's called the endoplasmic reticulum. We can abbreviate it the ER. It is found in both plants and animals. Okay, so the ER's job is to make lipids, those fats and oils. So the hydrophilic, hydrophobic, lots of, again, carbon, hydrogen, and oxygen only. These two, remember, have nitrogen in them. Sometimes we call it the SER or the RER. Okay, and we'll talk about why that is in here in a second. All right, so it says, it is much of the space in the cytoplasm is taken up by the ER, which has a folded system of membranes which form interconnected compartments in a cell. So it's kind of like the hallway in your house or your apartment complex or the school that connects all the classrooms. So it's a hallway or passageway. It provides a large surface area in which chemical reactions can take place. I know place got cut off by my hole puncher, sorry. Contains enzymes for all of the cell's lipid synthesis. So to make, synthesis means to make, and lipids. So it's the fats and oils that make up most of the membranes of the cell. When ribosomes, which we just talked about, produces proteins, are found on the surface, it's called the rough ER or the RER. So rough endoplasmic reticulum. Or if it does not have the ribosomes, it's called the smooth ER or the SER. The functions as the delivery system. I like to compare it to Amazon. Amazon's delivering a lot of packages in the world right now. So it's the delivery of the raw materials to the ribosomes for protein synthesis. So if we look at our diagram, all of this, these little surface areas right here, here's a smooth, and then this one has little bumps on it, if you can see it, is the rough ER. So here we've got the rough ER, and here we've got the smooth ER. Does not have ribosomes on it, but they're connected to the nucleus. And again, the ribosomes are made in the nucleolus. They leave through a pore, and they attach to the ER sometimes. So endoplasmic reticulum is, again, very large, much of the surface, much of the space of the cell, okay? And it makes the lipids, the fats and oils. All right, the next one we're going to talk about are the cell's storage organelles. So storage is important. The first one is the Golgi apparatus. Both the plants and the animals have it. It's called the repackaging center. So it's going to take all of the raw materials that were made in all of these other organelles and repackage them into a form that the cell can use. Okay, both plants and animals have them. It's a series of closed, stacked, flattened membrane sacs, membranous sacs that receive newly synthesized proteins and lipids from the ER and distributes them to the plasma membrane and other organelles. Proteins come to, and then we can abbreviate this one as well, as the GA in a vesicle. That's a vocab word. I forgot to highlight it in pink. Let me do that now. So the vesicle, where'd my pink highlighter go? All right, just know that the vesicle is a vocabulary word. I had a highlighter here. Sorry, guys. Anyways, the pink highlighter disappeared. Okay, so anyways, vesicle is a vocabulary word. Be aware of that. Vesicles are just pinches off of the ER. Okay, and I'll try to find an animation to show you that. And it's kind of like the grocery bag. You go to Kroger, you get your groceries, you put it in a grocery bag, and you take it to your final destination. So it's just a pinch off of the GA. Okay. And once the GA has the vesicle, it modifies the proteins chemically and then repackages them into a new vesicle for final destination in the cell. So for example, in Columbus, we have a lot of factories that produce things. Like for example, on the north side, we have the Budweiser plant. On the west side, or east side, far east from Columbus, we have what's called the Velvet Ice Cream Company. And we have the Pepsi bottling company as well. So maybe some of your parents work there. That company is kind of like the GA. They make huge vats of beer. And then there's another company that takes that vat of beer and then distributes them into the bottles. So the rebottling process, or taking a big vat of ice cream and putting it into the quart-sized containers, that's kind of what the GA does. It modifies it and puts it into a form that the cell can use. Okay. The next one is one that will help you also identify a plant. It is huge in a plant. Both plants and animals have what are called a vacuole. A vacuole is just a temporary space for storing food, water, waste, enzymes, and other materials by the cell. It is a sac of fluid, so it's not empty. It has fluid in it, surrounded by a membrane. So it is going to be really large in plants because they can't walk to the pond or the drinking fountain to get water. They have to store a lot of water and things like that. Or there's only a certain time of the year that they can produce a lot of their, say, carbohydrates when the plant leaves are green. Right now, we're in the middle of autumn. So the trees aren't photosynthesizing. They don't have the green leaves. Those cells have died. So they store their food in these large vacuoles. Okay. And so if we look at our diagram over here, the vacuole is very small in an animal, but really large in a plant. It takes up a lot of surface area in the plant. So let's see, I don't know if they have it labeled on this animal one, but they're very tiny. A lot of times they look like a lysosome, or here's what's called a peroxisome. Here we go, secondary vesicles. But they're very tiny. You can't really pick them out. Maybe it's probably one of these. So really tiny cells. So here, I don't really see one on here. Maybe one of these would be like a vesicle in an animal. So a lot of times vesicles in an animal can be confused with the next one we're going to talk about, which is a lysosome. And if you see a lysosome right here, they're also in plants. Lysosomes are found in both plants and animals, but they're very important because they're like the stomach of a cell and they store digestive enzymes. So it's very important. They have a really, really thick membrane around them because they digest excess worn out parts and food particles and invaders such as viruses and bacteria. The membrane around the lysosome keeps it from destroying cell proteins. So it keeps it from allowing, say, the stomach contents, like that acid that's in your stomach, to getting onto other organs and digesting it. So it's really important. The lysosome is found in both plants and animals, and it acts like the cell, so it's the digestive center. Okay, so that's the end of page 2. All right, one more to go, and then we'll talk about the diagram. All right, so these are energy transformers. Remember, energy cannot be created nor destroyed, so we're not making energy, it converts energy. So the mitochondria is in both plants and animals, and its job is to convert the carbohydrates to usable energy. So eukaryotes have organelles that transform energy from the cell. The organelle in which food molecules are broken down into energy. The mitochondria has an outer membrane and a folded inner membrane to increase surface area where energy storing molecules are produced. Okay, so if we look over here, so they kind of look like this. You can kind of see that they have a folded inner membrane and an outer membrane. So here's a cross section of a mitochondria right here. Lots of folded inner membranes. So it's in both. Here's an animal one, and there's a plant one. Okay, so in both plants and animals. All right, the next one's really important because it's only found in plants and some things called phytoplankton. So if you know plankton from SpongeBob, that's a phytoplankton. He's green because he has chloroplasts in him. This is where carbohydrates are made. Okay, so those sugars and those starches. So carbohydrates. Chlorophyll, again, chloroplasts are not in animals. Organelles found in green plants and some protists called protozoans. They transform light energy directly into usable chemical energy and store that energy in the form of food molecules called sugars and starches. They contain chlorophyll, which is the green pigment that traps energy from sunlight and gives them their green color. Inside the chloroplasts are three major components. We have the grana, which are intermembranous stacks of sacs. So here's a chloroplast right here. These little things that look like coins, these are what are called grana, okay? And they are very important. The stroma is the fluid that surrounds the grana. So if I were to point to a space in between these, there will be fluid in here. And the fluid is what's called stroma. And then the thylakoid is the inner membrane that has the chlorophyll embedded in it and traps energy from sunlight and gives the plant the green color. So it is this right here that has the chlorophyll in it, and that is what is called the thylakoid. Okay, so plants have these. This is a chloroplast right here, but as you see, there's no chlorophyll or chloroplasts in an animal. Okay, so it's very important because this is where carbohydrates are made. Remember, animals have to eat their carbohydrates, so they eat plants to get those. And so again, important fact, this is where carbohydrates are made, and then the mitochondria then break down the carbohydrates into the energy. So plants have both of these, mitochondria and chloroplasts, where animals only have mitochondria. All right, then last one for storage is plastids. Now plastids are just small membranous sacs that contain extra storage. They contain things like starches or lipids or other pigments. So plants are not just green. So the pigments that like roses have that make their petals red, or other colors that are not just chlorophyll, are stored in plastids. So these are kind of like secondary storage. So like in your house, the primary storage, the vacuole, would be like maybe the refrigerator, but then a plastid might be like a cupboard or a drawer in your kitchen. So plastids are very small extra storage for only in plants. All right, and the last few, I'm just going to talk about cytoskeleton. We got 4 more. Almost done, I promise. All right, so cytoplasm. This is important because cytoplasm is also in prokaryotes. It's in both plants and animals. It is kind of a jelly-like fluid that surrounds all the organelles. So it's the material that lies outside the nucleus and surrounds the organelles. It's a clear fluid that's a bit thinner than toothpaste gel. So if you ever had Aquafresh toothpaste, it's like the white toothpaste, but it has like a strip of green and a strip of red gel. It's the texture of the jelly, the gel, the green and the red in toothpaste gel. And it's usually about half the volume of the cell, and it's important for chemical reactions like protein synthesis to take place in the cytoplasm. So cytoplasm is everything that surrounds all of the organelles, okay? And same in plants. So it's the stuff in the middle, cytoplasm right here, and prokaryotes also have them. And I'll try to get a diagram of a prokaryotic cell to help you visualize this. Inside of that cytoplasm, sometimes they have, well, most of the time they have a cytoskeleton. So it's in both plants and animals. It's found within the cytoplasm, and it's a support system of tiny rods and filaments called microtubules and microfibules of protein, okay? And they form as a network of the cell, and they act as a support scaffold. If you've ever walked down the street in downtown and you see the window washers, they're standing on a scaffold. Sometimes high schools will use a scaffold to make bleachers. It's just a support system for the organelles. So here is the diagram of the microfilaments and so here's the plasma membrane, here's the mitochondria, here's probably the ER, yeah, there you go, and the ribosomes are on it. But then you have the cytoskeleton, which are these little blue things that are kind of interconnecting. It's kind of like the safety net. These things are not free floating in the cell. They're held in place by the cytoskeleton, okay? And then sometimes we have two organelles that are in animals only that help with movement. So single-celled animal cells can move around, but they would never have both. They would either have one or the other. So you wouldn't have cilia or cilium, which is called one, or flagella or flagellum, which is one. So a flagellum is like a tail. Cilia are kind of like hairs. So you can have thousands of hairs on the outside of a cell, but you would only have maybe one or two flagellum. So cilia are short, numerous, hair-like projections from the plasma membrane. They move in a beating kind of like pattern that is coordinated, kind of like a stadium wave. If you've ever gone to a football game or a concert or a basketball game and someone in the stands up and moves their hands like that, kind of like a whip-like motion, but one moves and then the one next to it moves and then down the line. So that's the way cilia works. So on here, we got a cilium, the little projections that come from the cell membrane. Plants do not have them at all. Nope, no plant cilia. So cilia would be all over the outside of the cell. A flagellum's like a tail. So if you think of like a sperm cell have long projections that move in a whip-like motion, only about one or two per cell. Okay, so we've got those. We've got the cell diagrams, and hopefully you've got all the notes. Again, remember you should have paused in between. And then the last thing I want to go over, if I can find it, is the diagrams that I've also given you in, yeah, here it is, in the notes. So here's the answer key. You can pause and fill it out. So this clearly has a very large vacuole. So that is number 5, but smaller in plants. So here's the answer key. Microfilaments are part of the cytoskeleton. We got a centriole. We'll talk about those. Centrioles are for cell division. Cytoplasm, number 2. We got chromatin, which is that uncoiled DNA in the nucleolus. Nuclear envelope. The nucleus is number 4. Microtubules, again, part of the cytoskeleton. Vacuoles, the very large or smaller in animals. Lysosome is number 6. The plasma membrane is just inside the cell wall, and it's on the outside of the animal. Number 7, mitochondria is number 8. This would be the rough ER, number 9, because it has the little bumps on it. Golgi apparatus kind of looks like a stack of pancakes. Number 10, here's the cell wall, the extreme outside of the plant cell. Nuclear envelope or nuclear membrane, number 13. We got the nucleolus, which is the center of the nucleus. Chromatin, number 13, or if it's coiled up, it's a chromosome, and then that's the nucleolus. Chloroplasts right here in the plant cell. And microfilaments, again, are part of the cytoskeleton. Same with the microtubule. So number 15 is the cytoskeleton. And cytoplasm is the gel-like stuff in between. If you have any questions, email me. Thanks and have a great day, and I'll see you in Zoom on Thursday and Friday.